Biology with Dr. E.
We start with passive transport. Passive transport requires no energy input, and all types of passive transport require diffusion. Diffusion is the movement of substances from a region of high concentration to a region of low concentration. This is also described as movement down a concentration gradient. Three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. This figure illustrates diffusion in 3 steps. In the first step, a dye pellet is placed in a beaker of water. The dye pellet dissolves, creating a high concentration of dye around it. In the second step, the dye molecules from the pellet are diffusing into the surrounding water. Kinetic energy makes the dye molecules move and collide constantly. As a result, they move down their concentration gradient from a region of high concentration to a region of low concentration. In the third step, the molecules from the pellet have finished diffusing and are equally distributed in the solution. When the dye is evenly distributed, there is no net movement of dye because there is no more concentration gradient. The rate of diffusion is influenced by 3 key factors. The first one is concentration gradient. The greater the difference in concentration between 2 areas, the faster the rate of diffusion. The next is molecular size. Smaller molecules diffuse faster than larger molecules, and that's because they have greater kinetic energy than larger molecules. The third is temperature. Higher temperatures increase kinetic energy, which result in faster diffusion rates. The system reaches equilibrium when there is no net movement of molecules in one direction. As already mentioned, the plasma membrane is described as selectively or differentially permeable. This is because it allows nutrients to enter the cell but prevents many undesirable substances from entering the cell. The plasma membrane also keeps valuable cell proteins and other necessary substances in the cell but allows waste to leave. Basically, it regulates what enters or leaves the cell. The hydrophobic core of the plasma membrane stops diffusion and creates concentration gradients by acting as selectively permeable barriers. Lipid-soluble molecules and small molecules can cross the plasma membrane unassisted. This is called simple diffusion. Meanwhile, water-soluble molecules and large molecules require assistance crossing the plasma membrane. This is called facilitated diffusion. Movement of water across the plasma membrane is called osmosis. So what happens if the plasma membrane is severely damaged? Severe damage to the plasma membrane results in substances diffusing freely into and out of the cell, thereby compromising concentration gradients across the membrane. For example, patients suffering from burns lose fluids, proteins, and ions from damaged cells. These patients are at increased risk of dehydration. Let's review simple diffusion. In simple diffusion, substances diffuse directly through the lipid bilayer unassisted. Non-polar lipid-soluble substances or hydrophobic substances directly cross the lipid bilayer. For example, oxygen, carbon dioxide, steroid hormones, and fatty acids would cross the lipid bilayer relatively easily. In addition, small amounts of very small polar molecules like water can pass through the lipid bilayer unassisted. This figure shows simple diffusion in which lipid-soluble molecules directly diffuse through the lipid bilayer. Lipid-soluble solutes from the extracellular fluid move through the plasma membrane into the cytoplasm. The molecules dissolve in and diffuse through the lipid bilayer relatively easily. Next, we look at facilitated diffusion. This is transport across the plasma membrane with assistance of carriers or channel proteins. Facilitated diffusion can be divided into two types: carrier-mediated facilitated diffusion and channel-mediated facilitated diffusion. In carrier-mediated facilitated diffusion, substances bind to and are transported across the membrane by protein carriers. Carriers are transmembrane integral proteins that transport specific polar molecules such as sugars and amino acids that are too large for membrane channels. Binding of a molecule causes a carrier to envelop it and change shape. That results in a molecule being moved across the membrane. Carriers can be saturated when all the carriers are bound to the molecule being transported. In channel-mediated facilitated diffusion, substances are transported across the membrane through water-filled channels. Channels are transmembrane proteins with water-filled cores that transport molecules such as ions down their concentration gradient. Channels are specific based on pore size and/or charge. Two types of channels include leakage channels and gated channels. Leakage channels are always open and allow substances to leak into or out of the cell down their concentration gradient. Gated channels are controlled by chemicals or electrical signals. This figure shows two types of facilitated diffusion. In figure B, lipid-insoluble solutes from the extracellular fluid move through the plasma membrane using a carrier protein specific for that molecule. Binding of the solute causes the carrier to change shape as indicated here, which facilitates its transport into the cell. Figure C shows channel-mediated facilitated diffusion through a channel protein. Small lipid-insoluble solutes, mostly ions selected on the basis of their size and charge, may diffuse through the plasma membrane in this manner.